a brief sketch about the professor Camarillo Inico. He did his education of a PhD from Wayne State, Detroit, and he did his postdoctoral research on endocrinology in the University of California. He has several experience on the research, and as a researcher, he has five PhD students currently and four MS students. And he used to have a model to show exercise during pregnancy can substantially decreases the breast cancer development in female offsprings. And he has employed a combination of memory surgical procedures and memory epithelial cell isolation techniques to characterize the hormone receptor expressions in developing memory gland. His expertise in the development of cell culture and animal models to study breast cancer progression places him in an excellent position. So his position and honors is currently the associate professor in Department of Biological Sciences, Purdue University, and he has several membership in the Professional Society and Honors of Eminence. And of the most, it is he received the Engagement Award from Purdue College of Science. Towards the contribution to science, he has significant number of publications towards his research study, and development of these models provided a foundation for several interdisciplinary collaboration is his main target. He has a very interesting joint venture projects in cutting edge technologies to determine the impact of diet components and obesity on human architecture, growth, survival, and proliferation. These collective research efforts have placed him in the excellent position to better delineate the role of obesity in breast cancer. Now, I invite Dr. Camarillo to start his presentation.
Huh. Well, thank you very much for that, introduction. And I am very appreciative of the invitation to come and to share some of my work with you. I'm very excited about this opportunity and glad to, welcome interactions with the new colleagues. So let me go ahead and share my screen so I can, get started on my talk here. Okay, let me adjust here. Okay. So my lab has been interested in a lot of different things, over the years, but more recently, we're really interested in identifying these bioactive molecules from plant sources that have anti-cancer properties, particularly those that can be effective towards, an aggressive form of cancer, breast cancer, triple negative breast cancer. And so what I'm going to be talking about today, in this talk is giving a little bit of background about the triple negative breast cancer, the epidemiology, and treatments. And then what we know at this point about some, vulnerabilities that this, type of breast cancer can have. I'll then talk a little bit about, in a more historical perspective, just briefly, some of the major plant compounds that have been identified that have been really important in developing, chemotherapies and anti-cancer treatments. And then I'll finish my introduction by discussing a little bit this particular plant that we're interested in, Lipia organoides, and how it, our studies indicate that it can be a rich source of anti-cancer compounds. So I'll focus in on, what our ideas are at the moment regarding our work, and then what our goals, more immediate goals have been. And so I'll then share with you what we've learned over the past couple of years on the characterization of the Lipia organoides on the viability of cancer, triple negative breast cancers, and, some of the mechanistic insights, in this process. And then I'll conclude by giving some, overall, the perspectives on our work and what our future directions, are aiming towards. So as an introduction to triple negative breast cancer, the epidemiology and treatment. So breast cancer is, worldwide, right, is a leading cause of cancer-related incidences and deaths. So what we see here is the number of cancer cases that occur clinically across the world, and this is in consideration of both sexes and all spectrum of ages. And you can see that breast cancer accounts for 11.6% of all new cases of cancer worldwide. And breast cancer is also a major contributor to cancer mortalities. Again, when we consider both sexes and all types of cancers across all ages. So breast cancer is a major cause of cancer-related deaths worldwide. It contributes to 6.6% of cancer-related deaths. So what's of particular concern, right, is our continued, projected incidences and mortalities, associated with breast cancer. So it's predicted that in the next 20 years, the incidence and mortality associated with breast cancer are going to increase by 50%. So there's a lot of efforts that are underway towards, identifying better, therapies and treatments for breast cancer. So one of the challenges of breast cancer is that it's a heterogeneous disease. So that is, when a woman presents with a tumor, that tumor is actually made of several different types of cancer cells. And this makes it very difficult, to find a treatment that works on all the different cancer cells in the tumor. So when towards understanding the origin of this heterogeneity, it's important to look at the structure and the cell types within the normal gland and where this heterogeneity originates from. So this is a picture of the structure of the mammary gland with the two major components, mainly adipose tissue and then an epithelial component. So the epithelial components ultimately produce milk during lactation, and these epithelial cells are susceptible to transformation for, the formation of breast cancer. If we take a closer look at the structural, cellular components of the epithelia, we find there's two major types of epithelia. Okay? So there's the luminal types. So these line the make up the inner lining of the alveolar structures and the ductal structures. So these epithelia make milk, and then they're transported through the ductal system, with eventually milk leaves the breast. Right? So the other main type of epithelial cells are basal. So these basal type cells are more contractile in nature, and when they contract, they help move the milk, through this ductal system. So there are specific markers that are associated with these different types of epithelia. And people have really isolated the difference, dissociated the cell types from the mammary gland, and then dissociated three major categories of cells that exist in the epithelia of the mammary gland. And these can give rise to, the major types of breast cancer. So the first type is derived from basal cells, and they're rich in stem cells. And these stem cells are important for giving rise to all other cells in the mammary gland, and they're important for repopulating, the mammary gland. The other subtypes arise from the luminal cells, and they come in the hormone receptor negative or hormone receptor positive types. Again, so basal cells are associated with a stemness of the mammary gland. The hormone receptor negative cells that are luminal are associated with high proliferation of the mammary gland. Hormone receptor positive cells are generally associated with a more differentiative type, of cell. And these are resistant, to transformation, but it does occur. Okay? So these different populations give rise to different types of breast cancer, which are associated with different aggressivenesses. So in this table, we see the different major types of breast cancer that have been identified, clinically. And we see here that the majority of these types, the luminal and then the normal like, are hormone receptor positive. And so these generally are more susceptible to therapy treatment. However, the breast cancers that arise from the basal cells are hormone receptor negative, and these are known as triple negative breast cancer cells, and these are much harder to treat. So when we look at the cases that arise of breast cancer at diagnosis, we see that the majority of the cancers are hormone receptor positive, and these are more treatable, either normal-like, luminal A, luminal B, or HER2 receptor. About 12% of breast cancer cases and so triple decreased survival rate compared to the other types of breast cancer, hormone receptor positive breast cancers. They have an increased risk of death within the 2 years of diagnosis, and they have a higher rate of relapse within 3 years with increased metastasis or spreading of the cancer from their initial site. When women are present with this metastatic type of triple negative breast cancer, their survival usually averages about 6 months, about half a year. So this triple negative breast cancer is a very aggressive form of the disease that's resistant to therapies. So there's a couple of different risk factors that are associated with triple negative breast cancer. So studies in the US have shown that African American women have double the risk for triple negative breast cancer compared to white, Latina, or Asian Pacific Islander, groups. Also, there's specific gene mutations that are associated with higher risk of triple negative breast cancer. So in particular, especially the BRCA1 genes, which are associated with DNA repair. When these genes are mutated, they're associated with an overall risk that's much greater for triple negative breast cancer, up to 16% overall risk. Excuse me. So other risk factors associated with triple negative breast cancer are the use of oral contraceptives before the age of 18, a premenopausal status, that is, women under the age of 40 have an increased risk of hormone negative breast cancers compared to older women. And women that are obese have a more than twofold overall risk, higher risk of triple negative breast cancer compared to women that are not obese. Some good signs or perspectives are that breastfeeding does prevent triple negative breast cancer associated with a reduction in TNBC incidence. So for all women, if they breastfeed for at least a year, they have a 31% reduction in triple negative breast cancer. And in particular, African American women benefit from breastfeeding. If they breastfeed at least, 6 months, they have 82% lower risk, so substantially lower risk of triple negative breast cancer. So what are the treatment regimens at this point that are associated, connected with triple negative breast cancer? Well, in general, there's different combinations of chemotherapies that are used to initially reduce the size of the tumor, and then this is followed by tumor resection, through surgery. So the two major categories of chemotherapy treatments, include, anthracyclines or taxanes, which result in a pathological complete response of 30 to 40%. When carboplatin is added to this regimen, then you get a higher level of the pathological complete response. If we consider the overall responses, in addition to the complete response, this regimen results in a response rate of about 85%. The 3 years of disease-free survival rate of these treatments is 60 to 85%. So unfortunately, a good portion of patients that undergo these treatments experience recurrence rates. So recurrence at different sites from the original point of breast cancer. So about a third of these patients. And when this recurrence rate occurs at distant sites, then the overall survival of these breast cancer patients that are metastatic is about a year. So these treatments, are not, as effective as we would like them to be. So when the cancer recurs, okay, usually what happens is that, surgery is initiated and then chemotherapy is given to patients with the post-recurrence, breast cancer. And in this case, the gemcitabine or ceftazidime and taxanes or platinum-based drugs are used. So each of these drugs, target different mechanisms for killing the cancer cells, either intercalating into DNA to stop the cell division, or direct interaction with the mitotic machinery, or increases in reactive oxygen species to promote cell death or apoptosis. So those are the general mechanisms of these chemotherapies. So unfortunately, the chemotherapies are associated with some severe side effects, including nausea, vomiting, neuropathies, anemia, and extreme fatigue. So the goal of the treatments currently is to identify treatments that are more effective, especially against triple negative breast cancers, and reducing the side effects. So finding things that can specifically target these aggressive cancers. So at this point, I'd like to go over some of the things that we know about triple negative breast cancer vulnerabilities. What are things that we can target? So in addition to DNA replication and mitotic, mechanisms, what are other things that we can target that would be helpful against triple negative breast cancers? And one of the things that has been identified is that there's a series of proteins in triple negative breast cancer cells that are in the category of this NF-kappa beta signaling that have been shown to be increased in the amount in triple negative breast cancer cells and increased in their activation. So early studies by Nikshatri et al. Showed that the NF-kappa beta signaling molecules are increased in breast cancer cells that are triple negative or hormone receptor negative in their status. So this data indicates, several different breast cancer cells that were used, right, to measure their NF-kappa beta amounts, and cells that have hormone receptor positive status have very low amounts of NF-kappa beta, while cells that have the receptor negative status have increased higher levels of NF-kappa beta. So NF-kappa beta is a transcription factor, and it binds to DNA and it activates several genes that are associated with survival and disease progression. So here's a cartoon that really summarizes how NF-kappa beta system works. So NF-kappa beta signaling within cells usually exists in two different forms of complexes: a complex 1 that's associated with the plasma membrane, and then a complex 2, which is activated when the receptors are internalized. So each of these systems are triggered when this TNF alpha molecule binds to its specific receptor. So in cancer cells, this complex 1 component are highly increased in amount and their activity, and this is predominant over the complex 2. In normal non-cancerous cells, usually the complex 2 predominates, and this TNF alpha signaling usually leads to cell death. However, in especially triple negative breast cancer cells, this system is activated which eventually leads to the activation of NF-kappa beta. So this protein complex, P50 and REL-A together, are known as NF-kappa beta. And this system can be activated. And when this NF-kappa beta enters the nucleus, it activates genes that are associated with cell survival and cell cycle progression. Okay? In addition, there's genes that suppress this complex 2 and suppress the cell death mechanisms. So when this system is activated, these proteins that are associated with the receptor activate kinases that in sequence phosphorylate this protein that's an inhibitor of NF-kappa beta. And when this protein is phosphorylated, it's marked for degradation. Okay, so under normal circumstances, this inhibitor of NF-kappa B, right, is associated with it and prevents its translocation into the nucleus. But when this is degraded by this complex 1 activation, this NF-kappa B is free to go into the nucleus and then promote cell survival. Okay. So at this point, we can say that triple negative breast cancers are often display aberrant expression of NF-kappa beta signaling proteins, and that targeting this system has been shown to reduce proliferation, invasiveness, and induce apoptosis in triple negative breast cancer cells. Okay. So in addition to dysregulation of NF-kappa beta in triple negative breast cancer cells, recent studies have shown that there's an extensive amount of metabolic reprogramming that occurs in triple negative breast cancer cells. And it's interesting because this isn't just one event, but it's a series of events that occurs. And it essentially creates new avenues for these aggressive cancer cells to really use additional substrates for producing energy and promoting growth in these cells. In normal cells, the predominant substrate, right, for producing energy is glucose. Okay, but in triple negative breast cancer cells, there's increases in mechanisms that allow these cells to use different precursor substrates to produce energy. So this chart summarizes several of these different metabolic reprogramming steps that lead to aggressiveness in triple negative breast cancers. So one of these is increased fatty acid oxidation. So using fats as a source of energy to promote ATP formation. And these also are a source of increased membrane lipid synthesis, which is important for rapidly producing, increased numbers of cells. The other thing that has been, for many years has been shown to change in breast cancer cells, and this seems to be an early stage of breast cancer progressiveness towards aggressiveness, and this is known as the Warburg effect. So essentially, there's an increase in aerobic glycolysis for the production of energy in these cells. So normally, this glycolysis or the breakdown of glucose into substrates that can be used for energy. Usually, this doesn't require oxygen, but even in the presence of oxygen, breast cancer cells increase their breakdown of glucose and other substrates to promote this glycolytic process for production of energy, which seems to be a first step towards the progression of cancer. Okay. We also see that in the breast cancer progression, right, there's an increased uptake and use of different substrates that feed into this tricarboxylic acid cycle, okay, that's linked with the mitochondrial activity and increased energy production. Okay. So once glucose in normal cells is broken down, right, its intermediates feed into the TCA cycle, and then eventually into the electron transport chain. And this is a very highly efficient way to produce large amounts of ATP. Well, breast cancer cells, again, increase the utilization of different substrates to feed into these mitochondrial systems. Okay. So one of the different sources of precursor molecules is an increase of overall several different types of amino acids that are used as a carbon source in addition to glucose to feed into mitochondrial mechanisms to produce in large amounts of ATP. And so in particular, aggressive cells and triple negative breast cancer cells use the amino acid glutamine as a precursor for producing energy. Okay, so they increase the shuttling of glutamine, this amino acid, into the cell and increase the utilization. So it's been shown that aggressive cells are addicted, to glutamine. And so when this happens, more substrates are fed into the electron transport chain, which leads to oxidative phosphorylation. And again, this involves a series of protein complexes in the mitochondria that produce large amounts of ATP. And it's been shown that when you inhibit oxidative phosphorylation, it can sensitize triple negative breast cancer cells to chemotherapies. So more recently, it's been, thought that these 2 points of metabolic dysregulation in cancer cells could be an important point for targeting, metabolic reprogramming in breast cancer cells. So it could create, development of agents that attack these points, create a new class of anti-cancer therapies that target metabolism. So this targeting of the metabolism may sensitize, the triple negative breast cancer cells, not only to chemotherapies, but agents that affect NF-kappa beta inhibition. Okay. So now I'll talk about some short perspectives on how plants and their bioactive compounds have been important in developing cancer therapies. So in the United States, right, the first record of the use of plants as a source of molecules, right, for treatment of several different, diseases or medicinal purposes was first recorded by use of the Native Americans and their use of the Podophyllum peltatium roots. And these extracts were prepared and they were used as cathartics, anti-parasitic agents, purgatives, and anti-cancer agents. So around the world, right, it's been well documented that different populations have used plants for a variety of medicinal purposes, right? But especially in the early or the mid 1800s, there was a series of, experiments that began that started to identify the bioactive components, from these plants. So one example that's tied to this podophyllum is early experiments by John King, which published the first papers showing that they were able to extract resins from this podophyllum that had bioactive, components. Okay. Later studies by Podwastowski showed that they were able to specifically isolate and crystallize the specific component that's responsible for the bioactive activity, of this, resource of podophyllum. Okay. And later on, this toxin, this podophyllitoxin, as it was named, was shown to be particularly effective in killing, cancer cells in vitro and in vivo. Okay. Ultimately, further studies by Stalin and Wartenberg developed derivatives of this podophyllitoxin, okay, which were identified as epotopside and tenopside. Okay, and these are, effective anti-cancer agents that are a class of what's known as the topoisomerase inhibitors. So they inhibit DNA. So they inhibit DNA structure. Okay. Are there questions? Are there questions? Yeah. No.
No, sorry, please proceed, proceed.
Oh, okay. Okay. Yeah, no problem. So then this is one of the initial major perspectives on the development of chemotherapies from, plant sources. So around this time, right, there was other, examples of plant-derived compounds, right, that resulted in anti-cancer agents, particularly the study of the plant C. rosaceus, okay, led to, the isolation of components from this extract that, specifically, were identified as the vinca alkaloids. And so these are anti-mitotic agents. So they disrupt the machinery that's associated with cell division. So around this time when these molecules were identified from this plant source, the National Institute of Health, right, established the National Cancer Institute, and its main goal was to begin large-scale screening of natural extracts, right, to identify bioactive compounds that could lead to anti-cancer agents. And then this work, additionally led to the identification of camptothecin and taxanes at Research Triangle Institute. And again, these are important molecules that inhibit, cell division and promote cell death. So more recently, there's various other plant-derived, compounds and extracts that have been shown to have anti-cancer activity. In particular, the molecule resveratrol, which is present in the skin of grapes, has been shown to have anti-tumorigenic activity. Also, extracts and molecules from sources of the nascopine, curcumine from the plant turmeric, and parthenolide have also been a source of anti-cancer, activities. And the interesting thing is that each of these more recently discovered molecules have been shown to have multiple effects on mechanisms in cancer cells to suppress their proliferation and activities. And so collectively, these studies really support the continued, interest in medicinal plants as a rich source of new classes of compounds for cancer drug discovery. So at this point, I'll focus in on our interest in the Lippia organoides, or it's known as also a mountain oregano. So Lippia organoides is found predominantly in, Central and South America. It's the mountains of Central and South America. And there are over 200 species of Lippia in general. Okay. And here we show that the different species of Lippia have been associated with different activities that are connected with anti-mutagenic, anti-inflammatory, and anti-cancer properties. And so these different species, of Lippia have been tested in colon cancer and liver cancer. So in particular, the Lippia organoides, right, when these extracts are analyzed, it's been shown that several components that were identified within Lippia have been are connected with the inhibition of NF-kappa beta signaling. So when Lippia organoides components have been identified in the past, right, this is a list of some of the major components in this Lippia organoides, and these have been tied to the regulation or suppression of NF-kappa beta system in several different cell types. So at this point, we can say that the aberrant and pro-inflammatory NF-kappa beta signaling, glucose utilization, and metabolic reprogramming are hallmarks, characteristics of triple-negative breast cancer cells, and targeting these pathways can be new viable routes, for effective therapeutics. Lippia organoides contains multiple components which are known to inhibit NF-kappa beta signaling. And at this point, we thought, well, this may be a good candidate for testing against triple-negative breast cancer, which, at the beginning of our studies had not been tested yet, okay, in this context. So at the beginning of our studies, we hypothesized that this species of Lipia, the organoides, the extracts would inhibit NF-kappa beta and produce suppress proliferation and cell death in triple-negative breast cancer cells. And so our studies focused on characterizing the actions of Lipia organoides in triple-negative breast cancer in vitro and gaining insight into the mechanisms of this extract in triple-negative breast cancer cells. So we'll move on to some of the major results that we've come across in our initial studies in characterizing the actions of Lipia organoides. So Lipia organoides, right? A specific chemotype that we use is L42. So this L42 and Lipia organoides are going to be used interchangeably in these subsequent slides. Okay, so this Lipia organoides chemotype was tested in different triple-negative breast cancer cells. So the MDA MD231 is a receptor negative representative of triple-negative breast cancer. And then the other cell type that we used is MCF10A, and this is representative of normal mammary cells. You know, ultimately, we used a number of different triple-negative breast cancer cells and normal cells. Okay, but these two charts represent what we saw in these broader categories of cells. So essentially, when we treated these cancer cells with increasing amounts of Lipia for 1, 2, or 3 days, we saw that there was a massive decrease in the number of cells with the increases in this Lipia treatment and increased time. Okay, so this Lipia L42 had, you know, a dramatic effect on the reduction of cells of the triple-negative breast cancer type. Now, when we treated the normal-like cells with the Lipia, we saw a much different curve that were generated over these range of Lipia treatments and times. We saw that, and particularly at the lower concentrations, there was very little effect that the Lipia had on these normal cells. Only at the highest concentrations did we see some kind of a reduction of cell numbers. Okay. So we wanted to find out what was causing the reduction in cell numbers. Usually a reduction in cell numbers is a result of increases in cell death or the suppression of the cell division. Okay. And so we look specifically at different methods to measure the cell death or apoptosis in our system. So essentially we used a FACS analysis to identify cells that were alive, that were undergoing early, stages of cell death or later stages of cell death. And in these three categories, we see cells that were not treated with Lipia or triple-negative breast cancer cells that were treated with increasing concentrations of Lipia. When this data is transformed, we see that there's an increased amount of cellular apoptosis or cell death in cells that are treated with Lipia, right, compared to cells that are not treated with Lipia. And an important thing to note here is that this type of cell death apoptosis is programmed and intentional. It's not a toxic type of cell death. And so this is what's occurring in these cells as a response to Lipia. And we did not see an increase in necrotic cell death in these cells. So this was very low. And this necrosis is a reflection of a very harsh toxic type of treatment of cells that usually is non-specific and can kill normal cells or cancer cells. And ultimately you want treatments that are only going to target, right, these breast cancer cells. And so this is a good sign that this Lipia treatment promotes this very intentional apoptosis cell death form, in triple-negative breast cancer cells. So knowing that L42 induces apoptosis, we wanted to look at specific markers that are associated with apoptosis. So very briefly, we measured specific levels of proteins, caspase 8 and PARP, that were associated with, that are known to be associated with increases in apoptosis. Okay, and essentially, when measurements of protein levels were transformed. We saw that lipia treatment at short-term or long-term, well, actually this isn't very long-term, but longer-term treatments had a dramatic increase in both of these proteins, caspase and PARP, right, which are associated with apoptosis. So this is a, foundational mechanism insight that accompanies, this promotion of apoptosis. So at this point, we could say that lipia induces significant and sustained loss of viability in triple negative breast cancer cells. So this is a selectivity that was not present towards normal mammary cells, and that the cell death that occurred in these triple negative breast cancer cells was associated with apoptosis, and particularly connected with caspase activation. So this is an overall scheme of what we're going to be filling in along the way in this talk on the actions of lipia in triple negative breast cancer cells. And what we know at this point is that this lipia results in this caspase activation, which leads to apoptosis. Okay? So what we want to know is what are the mechanisms that are altered by lipia that lead to this ultimate, cell death. So that was the focus of our next aim of studies, where we wanted to gain insight into, specific mechanisms of the apoptosis, triggered by lipia. In our first study, we showed that the lipia actually, in addition to apoptosis, it was able to suppress, cell division process or suppress the cell cycle. This data shows a measurement, the use of FACS analysis to gain insight into the different major stages of cell division or the cell cycle, which are represented by, the G0, G1 interface. So this is the beginning of the cell cycle. When cells progress in their cell division, they enter a growth 2, the second growth phase, and they go through a DNA synthesis phase, which eventually leads to cell division. So you can see here that cells that were not treated with lipia are going through these different stages of the cell cycle. Okay? And cells that are treated with lipia have a reduced amount of cells that are in these cell cycle progression phases. So when this data is transformed, we can see that there's a much reduced amount of cells in these latter stages of the cell cycle, and more cells are caught at the beginning of the cell cycle. So they're frozen in this initiating part of the cell cycle interface. Okay? So the next step was to really quantify proteins that were associated with cell cycle arrest. And we measured individually, through Western blot analysis, levels of proteins that are associated with cell cycle. These are cyclin D1 and CIAP2. And when this data is transformed, we see that lipia treatment, right, substantially reduces, okay, the amount of CIAP2 and cyclin D1. So this is known to suppress the cell cycle and actually inhibit the inhibitor of apoptosis. So this is promoting the cell cycle and promoting mechanisms that ultimately lead to apoptosis. Another protein that we specifically measured was this RIP2, so this receptor interacting protein. So we measured this, so this is an initial event in NF-kappa beta signaling. And essentially we showed that lipia treatment at a shorter time period results in a substantial reduction in this RIP protein. So this lipia really suppresses the initial stages of NF-kappa beta signaling. So lipia not only suppresses cell cycle arrest, but it inhibits a major scaffold protein in the NF-kappa beta signaling. So now we can add some understanding of how lipia ultimately leads to apoptosis. So here's the NF-kappa beta signaling that in triple negative breast cancer cells promotes proliferation, cell cycle, right, and suppresses apoptosis. But in our studies, we honed in on RIP1 and CIAP2 and cyclin. We showed that Lipia can substantially reduce these proteins, okay, which ultimately are a reflection of the suppression of this NF-kappa beta system. So simultaneously, when you suppress this complex 1 system, okay, so this suppresses, leads to cell cycle arrest, but then it also promotes the activation of complex 2. Okay, and this complex 2 leads to the promotion of caspase activation and apoptosis. So you're having 2 major effects simultaneously by suppressing the NF-kappa beta. Okay. So we initially honed in on specific aspects of the actions of Lipia, but then we wanted to, further our studies and really use more broad scale methods, for really identifying the full landscape of effects of, the actions of Lipia. So one of the things that was available to us that we used was this quantitative proteomics. So this is a very useful method in a couple major fronts. First, it allows us to identify proteins in, very complex mixtures in cell preparations or, tissue preparations, and its sensitivity is very high. And so you can, identify even, proteins that are very low abundance. So you can really get an insight into the broad spectrum of, the mechanisms, in your system. And you can also identify molecules that could be potential new targets, for cancer, but it also gives you early insight into, off-target mechanisms that could lead to, side effects of the mechanism of action, of your treatment. So this next slide really summarizes the workflow of our proteomics and the reproducibility of our data that we generated in our proteomics. So essentially, cells, the MD231 triple negative breast cancer cells were treated with Lipia or not treated with Lipia for about 12 hours. These cells were isolated and then homogenized, and these homogenized samples were then separated into membrane soluble fractions, okay, and then membrane insoluble fractions. So these represent the 2 major components of the cell. Okay. Once these major components were isolated, then the cells from these were further purified, and then the proteins were broken up, right, by specific enzymes, the trypsin and lysosome C. Okay, so these enzymes break up the proteins into small peptides, and this is a really important, right, for ionizing these peptide fragments and then identifying them, using the tandem mass spec. Okay? So once these proteins are isolated and then they're trypsinized, right, then they're prepared through solid phase extraction and drying, and then they're used, resuspended and used for LC tandem mass spec. Okay, and this process essentially, ionizes these peptides. Okay, so they can be, more easily identified. So each of these peptides, from a protein is identified, and then these protein fragments, right, profiles, right, are used, with peptide databases to identify a list of proteins that are present in your samples. Okay, and then the advantageous thing of this system is that when these, spectra of these peptide fragments are identified, right, this program MaxQuant is used to assign a quantitative fluorescence, right, to these peptide fragments. And then this assignment of this peptide fluorescence to the spectrum of the fragments essentially leads to a quantifiable measure of these peptide fragments that when they're average gives you an estimate of the amount of a particular protein in these samples. And through this max quant, you can actually identify, right, the differences in the levels of specific proteins, across your different treatments. Okay. So once the list of differentially expressed proteins are identified, that list is fed into different software tools, the enrichment and pathway analysis, and this gives us insight into how these proteins are related and what their functions are. Okay. This data just indicates the consistency and reproducibility of our samples and analysis of our samples through this process. So this L42 samples represent cells that were treated with our Lipia organoides. Okay? And these are their control samples. And we can see here that from this map, when you look at the spectra from within these sample treatments, you see that the R² values are identical. And that's an indication that this is a high quality data that's consistent. Okay, and within the control samples, we also see R² values that are very consistent. And across samples, we see the much lower R² values. Okay, this shows that our preparation of our sample and our workflow is of high quality. Okay. So then the next step was to really create a list of proteins through this quantitative system that we could analyze and get some mechanistic insights from. Okay. And so essentially when this process was used to identify proteins, okay, in our system, we identified over 3,000 proteins across our replicates, okay. But through the filtering process, we used about 2,400 of these proteins for analysis. So the filtering process basically removed proteins that were not present across all our different replicates. Okay. So this LFQ, right, stands for label-free quantitation. So this again is the assignment of a fluorescence intensity to the spectra of these specific proteins. Okay, and these specific spectra so that we could, more easily compare the values and the quantity. They're transformed into log values of these LFQ, measurements. Okay, and then essentially there was a difference in these log values across the samples were quantified, and they were termed, the delta of the log 2 of the label-free quantitation of the particular proteins. And essentially, when we consider these values, okay, the proteins with a high, change across the treatments were used for our gene ontology analysis, right, to identify which genes or which proteins were most, changed in our treatments. And so the criteria for using these are proteins that were changed more than a negative 0.5 value or lower than a 0.5 value of the change of the log intensity, or higher than a positive 0.5 value, with a p-value of 0.05. So these data were fed into our software analysis to identify, the proteins that were differentially expressed upon our Lipia treatment. And these list of proteins were really evaluated in a couple of different contexts using the Go software or the gene ontology, software. They were used to really look at, how these differentially expressed proteins, were localized in different parts of the cell, right? And what the functions of these proteins were, okay? So this data represents a heat map. So the changes in the levels of the proteins, that occurred upon Lipia treatment compared to vehicles. And this red area of the heat map shows an increase in, the amount of proteins that were present across the samples, and the blue represents a decrease in the proteins that were present across these samples. So here we show predominantly all the cells or all the proteins, right? There's about 500 proteins that were down-regulated in response to Lipia, compared to control. And a vast abundance of these proteins were found to be associated with the mitochondria. Okay. So this is an interesting observation that this Lipia is predominantly affecting, mitochondrial function. Okay. So when we look at the enzymatic activities that are downregulated by Lipia, again, in this other perspective, we show that there's a lot of different enzymatic activities that are associated with mitochondria that are suppressed by the Lipia, by the L42. And this is associated with electron transport chain, NAD binding, or the oxidative reductase activity. Okay. So when we take a closer look, right, at the presence or the downregulation of proteins, right, in specific processes, we see that within the mitochondria, right, Lipia seems to really affect two major parts. One of that includes the tricarboxylic acid cycle. Okay, so the tricarboxylic acid cycle is a series of enzymes that's important for the production of substrates, right, that lead to the feed into the electron transport chain and increase ATP production. So in normal cells, right, a major precursor for the activation of the TCA cycle is glucose. Glucose is metabolized, right, and then its metabolite feeds into the TCA cycle, right, to produce things like succinate, okay, and other molecules that feed the electron transport chain, within the mitochondria to produce ATP. Okay, so as I mentioned before, right, triple-negative breast cancer cells have an increased metabolism of different substrate sources besides glucose, including fatty acids and amino acids. And these feed into the TCA cycle, right, that eventually leads to the activation of the electron transport chain and production of energy, ATP. Well, interestingly enough, from what you see here, right, the proteins that are part of the TCA cycle that are in blue are proteins that are downregulated, okay, by Lipia. And so we can see that Lipia results in the downregulation of several proteins. So it inhibits the TCA cycle at several different points. And in particular, it has a profound reduction in the complex of proteins that's known as the enzyme alpha-ketoglutarate dehydrogenase. And this is a rate-limiting enzyme in this TCA cycle. So again, this is a really important part of a suppression of the TCA cycle that's affected by Lipia. Okay. So another major part of the function of mitochondria that's affected by the Lipia is the electron transport chain. So the electron transport chain is a series of protein complexes, 1 through 5, that exist on the inner membrane of the mitochondria. And essentially, substrates from the TCA cycle feed into these different complexes, and they provide protons and electrons that are fed through these complexes. And this energy from these electrons causes the movement of protons from out of the inner part of the mitochondria to this intermembrane space. The accumulation of these protons then ultimately filters back into the inner mitochondrial matrix through this ATP synthase. So this is associated with this complex, and this is really important for producing large amounts of ATP. Okay, so what we found was that the Lipia, the L42 treatment, caused a substantial decrease in several of the proteins that are associated with complex 1, which is a rate-limiting step in this electron transport chain, or particularly this complex 1. And so it's interesting that the proteins that are part of this complex are actually, produced from the nucleus of the cell, and they're nuclear encoded and transported right to the mitochondria. So lipia may be affecting the expression, of these genes or the transport of these proteins into the mitochondria. Okay. So under normal conditions, when the mitochondria is very active, there's a large amount of negative charge within the mitochondria, and this membrane potential of the mitochondria is a very negative. Okay, but when you inhibit this electron transport chain, right, we anticipate that this reduction of this activity leads to a depolarization of the mitochondrial membrane potential. So in other words, this membrane potential or membrane electrical properties of this membrane is less negative than when it's in the active state. Okay. So we actually biologically tested the membrane potential of the mitochondria in response to the lipia treatment. And what we showed, so this is a treatment of the triple negative breast cancer cells in response to lipia, and this is our vehicle controls. This is our background fluorescence showing, the outline of the individual cells. And this is the fluorescence measurement of the mitochondrial activity. So we use this trimethyl rhodopsin. This is a fluorescent molecule that's very positive, and this associates with active mitochondria. So this fluorescence can be quantified, and under normal conditions, right, when mitochondria are very active, you can see that this increased activity of mitochondria can be localized, okay, in these highly active triple negative breast cancer cells. However, when lipia, is treated, exposed to these breast cancer cells, we see a substantial reduction, right, in the amount of the fluorescent dye that's in these cells. And this is a reflection, a reduction of the activity of the mitochondria. And this is a transformation of the data showing a reduction in the activity of mitochondria. So we've biologically, confirmed that this inhibition of the mitochondria by lipia does result in reduction of the membrane potential of the mitochondria. And so this mitochondrial reduction in membrane potential is associated with, the triggering of caspase-3 and apoptosis. Okay. So this seems to be having a dual effect. So in addition to inhibiting, right, mitochondrial activity, it seems that lipia also, through our proteomic analysis, suppresses enzymes that are involved in, amino acid metabolism. So I mentioned before that in aggressive cancers, right, the cancers upregulate, enzymes that can lead to the use of alternate precursors for the production of energy, namely, amino acids as carbon, sources for the production of energy. Okay. Well, we've shown through our quantitative proteomics that there's a numerous amount of proteins, right, that are downregulated upon lipia treatment that are associated with the metabolism of amino acids. Okay. So when we generated this list that was reflective of enzymes associated with amino acid metabolism, we actually wanted to gain some insight into the interconnectedness of these proteins that were downregulated. So we used a string analysis, right, to analyze these proteins. And basically what this is showing is that of these proteins that were downregulated, we show that these a variety of these proteins have a direct interaction with each other. So that's what these lines between these proteins mean. So they can form, pathways, right, that lead to the regulation of amino acid metabolism. Okay. The other thing that we show here is the localization of these proteins. And we saw that the vast majority of these proteins are present in the mitochondria. either in the mitochondrial matrix, as indicated in red, that shows that they're feeding into the TCA cycle, or the mitochondrial inner membrane. Okay, and this shows that they're more associated with producing substrates that are important for the electron transport chain. So another interesting thing that we found from our quantitative proteomic studies is that Lipia really suppressed this glutamine or glutaminase enzyme. Okay, so this enzyme is important in the metabolizing of glutamate to glutamine. So this is a major important amino acid for production of energy in triple negative breast cancer cells. So to confirm this quantitative proteomics finding, we actually use specific antibodies to measure differences in levels of glutaminase in triple negative breast cancer cells that were treated with Lipia compared to those that weren't treated with Lipia. And again, we saw a massive decrease in the amount of protein of this glutaminase. Okay. So not only does this glutaminase, right, is this involved in regulating amino acid or glutamine, processing, but it's also been shown that this loss of glutaminase can suppress triple negative breast cancer cell growth. Okay. So to summarize this section, okay, we've shown that the Lipia organoides can induce cell cycle arrest through inhibition of cyclin and CIP2 proteins. Okay. That along with these reductions, they can also reduce the RIP protein, which has an inhibitory effect on NF-kappa beta signaling. Triple negative breast cancer cells treated with Lipia also show a significant loss of enzymes involved in mitochondrial metabolism and in membrane potential. Okay. And this is associated with the triggering of enzymes, particularly caspase 3, that triggers apoptosis. So in addition to the Lipia's effect on the NF-kappa beta system, through these more recent set of studies, we've shown that Lipia can suppress different actions within the mitochondria, including directly reducing proteins involved in the TCA cycle, or reducing proteins, particularly in complex 1, that are involved in the electron transport chain, which is a major producer of ATP. At the same time, Lipia can suppress levels of the glutaminase enzyme, which is important in converting this glutamine amino acid, right, into glutamate. And this glutamate can serve as an important precursor for generating other amino acids, or it can be metabolized through the TCA cycle, right, to create energy or also to produce amino acids and lipids. Okay. So this is having additional effects, right, Lipia associated with mitochondrial action. Okay. And again, these can also lead to caspase activation and apoptosis and cell cycle arrest. So overall, what our studies have shown is that this, in this project, we showed that Lipia organoides can induce potent and sustained apoptosis in triple negative breast cancer cells, and there's more selectivity compared to normal cells, that the mechanisms of apoptotic action of the Lipia includes inhibition of NF-kappa beta signaling, halting mitochondrial metabolism, and mitochondrial membrane depolarization, so altering membrane amino acid metabolism as well. And so these studies support that NF-kappa beta and mitochondrial metabolism could be a new source of targets for development of new classes of anti-cancer agents that target metabolism. Okay, and that simultaneously targeting these areas of multiple vulnerabilities can lead to sustained apoptosis of highly aggressive triple negative breast cancer cells with a reduced effect on normal cells and reduced toxicity. Okay. We also have shown that the vast power of the omics technology to more rapidly identify, complex changes in the cells in response to different treatments, and they can identify, expedite the identification of bioactive molecules from complex mixtures and identify their mechanism of action. Okay. Again, identifying new targets, for breast cancer. And this has important also implications for identifying off-target effects. So in the earliest stages, these broad scale methods can give insights into, potential negative side effects of treatments that are being developed. So ultimately, these give rise to a reinvigorated, interest and support for the investigations in medicinal plants as a rich source of new molecules that can suppress cancer, particularly those that, target metabolism. And particularly, we're interested in this, the actions and the molecules, associated with lipia. Okay. So our future directions, to end this talk, the future directions are going to focus on how these bioactive components in lipia. So we've actually done some studies that identify, highly abundant molecules that are present in lipia, but we want to better understand how these components of lipia interact, okay, with, proteins and other molecules in breast cancer cells that lead to these mechanistic changes in the mitochondria and F-kappa beta that lead to the killing of triple negative breast cancer cells. Of course, we also want to lead into studies in animals that show that lipia can suppress, triple negative breast cancer cells in vitro. We've already done some studies where we test the toxicity of lipia in normal animals. So in animals without tumors, we've shown that lipia does not cause toxicity at high levels of lipia treatment. They don't cause toxicity. And then the other thing that we want to do, which we've actually gotten some data just recently, we want to really identify, the gene regulation effects of lipia in these triple negative breast cancer cells, particularly genes that are regulated, that are associated with complex 1 and NF-kappa beta. Okay, and again, we want to use these broad scale. We've used RNA-seq technology to look at these early stages of changes in gene expression that could lead to changes in protein expression and give us a better insight into, the important mechanisms and targets, of these lipia, actions. So I'll end my talk at this point, and I'll be happy to entertain any questions at this point. But before we do that, I'd like to reference or acknowledge, right, as the collaborators and people that have worked with me and worked within my lab. Predominantly, first and foremost, I'd like to thank Vishak Raman, a graduate student in the lab. He graduated last year, and he did, a majority of the work that I presented. Also assisting in this process, towards the transformation and analysis of data and propagation of cells with the undergraduates, Rodrigo Ferreira, graduate student Leksha Mittal, and then Ivy Camarillo. And then also I want to thank, Raji, who has been very insightful in this whole process and was very generous in connecting me with this opportunity to share my research today. And also very grateful to Dr. Morris Levy and Maria Levy, who connected me with my collaborators in Colombia that are the source of this lipia extract. So Dr. Fuentes and Stasenko were collaborators that provided background knowledge and as a source of this molecule. I also want to thank our collaborators at Purdue who helped perform these proteomics, quantitative proteomics analysis that led to our findings. Okay. So at this point, I'd like to very much, open to questions about any aspects of this, talk. Okay. So I see this first one. So are you treating the patients with L42, and what is this effect of L42 that gives the desired results? Okay. So at this point, we have not treated patients with L42 directly. And I will say that, the big challenge in treating, animals or patients directly with this compound is that, sort of better understanding of how this compounds are metabolized in vivo. Okay. And so, like I said, we have done some initial in vivo state studies injecting animals without cancer with this Lipia, and we've shown that it does not affect the animals. The animals, are definitely, responsive, and they don't show any signs of toxicity. And we've actually analyzed the mammary tissue in these animals, and they show that the structure of the mammary gland is not affected, and it doesn't undergo apoptosis. So it doesn't seem to be toxic under normal conditions. Now, other studies, so looking at other impacts of different species of Lipia, have been done, in vivo in animal studies, and they've shown that injection of Lipia through intraperitoneal venues does seem to generate, actions of Lipia, in other, disease regards. So we're very, enthusiastic about these possibilities. So in addition to, the IP venue avenue of introduction of this Lipia into animals, another possibility that we've been considering, right, is introducing these Lipia components into these hydrogels, which can be used to be implant into tumors, especially tumors that are hard to really treat and resect surgically. And there's evidence out there showing that that these different, forms of encapsulating, Lipia components have been effective in not only stabilizing Lipia, but causing its, timely release, into the environment. So that's an avenue that we could, really pursue that could, address, the possibility that, Lipia could be metabolized, once it enters into the system. But there's, like I said, out there in other studies that are not related to cancer, there's promising results showing that it can be given, in vivo. Okay. Yeah. Okay. Other questions?
That was a very good presentation, Professor Camarillo. So, on the growth of the cell structure, uh, whether do you estimate the structural growth of the cell periodically, or how does your cell growth is measured?
How does a cell, what's the characteristic of the cell inhibition over time? Is that your question? Yeah. Yeah. So this is interesting, right? Usually with different therapies, I could show this data, but I'll just mention it, right? One of the problems is when you treat cells and cancer cells with any kind of agent, right? There's a response that occurs, okay? But then eventually the cells, return, right? Once that reagent is removed. So interestingly enough, we've done some in vitro studies where we have kept, right, from the results that I've shown you at this point, they're reflective of treatment of these breast cancer cells in vitro, where the Lipia is kept on these cells over 3 days. Now, to test whether Lipia can have, effects that are ongoing in its absence, we actually treated triple negative breast cancer cells with Lipia for 1 day and then changed the media, to the standard media without Lipia, and then kept track of the cell viability over a period of, 3 days and longer. And what we shown was that even if you remove Lipia from the media, right, the actions, the anti-proliferative and pro-apoptotic actions of Lipia are sustained. Okay, so this means that whatever, the activities of lipia that occur initially when the lipia components bind to their targets in the triple negative breast cancer cells, those are permanent and they're irreversible. Okay, so once those actions occur, right, they're sustained and they continue to kill, cancer cells even in the absence of the continued treatment of lipia. So this, in addition to lipia's selectivity towards, breast cancer cells and not normal cells, this is something that, is really intriguing and piques our interest in the further study of this extract as a source of, molecules that could really, address this triple negative breast cancer.
Thank you.
Other questions?
Dr. Camarillo, good
Yes.
evening, good morning.
Good morning.
I have one general clarification.
Yes.
What is the current status with respect to targeted delivery of chemo medicines to the cells?
Could you repeat that? What is the?
What is the current status of the development on targeted delivery of chemo medicines to the cancer cells?
Yeah. So what's okay. So what's the current status of targeted delivery of the chemotherapeutics? Well, so this is interesting. So there's a lot of different, avenues, right? So I mentioned that the implantation of these hydrogels, right, that are non-toxic, themselves, is an avenue that you can really target, breast cancers. Another avenue that people have been, studying is the encapsulation, of molecules into nanoparticles that, again, preserve their, structure and protect them from clearance from the body and protect them from the degradation. So there's different types of nanoparticles, that are produced, and essentially, whatever reagents that you're studying can be associated with and loaded into nanoparticles that can be introduced into, the body that increases the viability or the stability of these molecules. Okay. The other thing that's been really studied, and particularly here at Purdue, is the ability of conjugating anti-cancer agents to specific antibodies that target breast cancer cells. Okay, and so at Purdue, there's this researcher, Phil Lau. Okay, and he has developed this entire system. He founded a company, the Endocyte, that established this technology that basically is based on the specific delivery of molecules to cancer cells based on the overexpression of folate receptors on cancer cells. So cancer cells are very highly metabolically active, right? And they depend on high levels of folate to come into the cell, right? To then promote all types of metabolic reactions, right? So he found out that when you use an antibody to the folate receptor, it can specifically seek out and bind to folate receptors that are on the cell membrane, and those receptors are enriched on cancer cells. Excuse me.
Yeah.
And different studies, from his lab and collaborations have shown that you can conjugate different reagents, okay, and even fluorescent imaging agents to the folate antibody. So this folate antibody, right, when it seeks out the cancer cells specifically, right, it brings with it, immunofluorescence dyes or, different types of anti-cancer agents that it can be concentrated in the cancer cell and not so much in the normal cell. And this has shown, considerable promise in clinical trials, the various types of methods that are based on this technology. So that's another promising avenue for, targeted delivery of agents.
So, Mr. Camarillo, one more clarification.